Action.
Have you ever wondered how a simple thought to move your arm or leg becomes the actual motion? There's actually a lot that goes on beneath the surface of our body to make muscles move or contract. Let's take a closer look. Every muscle contraction begins with an action potential or nerve impulse from the brain. When the action potential reaches a synaptic knob of the neuron, it opens calcium channels in the membrane of the neuron. This allows calcium or calcium ions to move into the cell. When the levels of calcium increase within the neuron, the synaptic vesicles begin to get filled with acetylcholine, a neurotransmitter. Once the synaptic vesicle is completely filled with acetylcholine, it exits the synaptic knob through exocytosis. Once the acetylcholine leaves the synaptic knob through exocytosis, it enters the synaptic cleft and begins to move towards the motor end plate, or the motor neuron, and all the skeletal fibers it stimulates of the muscle. The acetylcholine is picked up by the acetylcholine receptors that contain the sodium and potassium channels. These channels open and the sodium ions diffuse into the cell, which make the membrane more positive. This is also called depolarization. This depolarization can cause another action potential on the sarcolemma or cell membrane if it hits threshold. The acetylcholine is recycled back and broken down by the enzyme acetylcholine esterase. This action potential travels along the sarcolemma and eventually hits a T-tubule or transverse tubular, which is located in a fiber within the fascicle of the membrane. This releases more calcium from the terminal cisterine of the sarcoplasmic reticulum. It was being stored by calsequestrin. This released calcium binds with troponin on the thin filament and pulls tropomyosin off the active site found on actin. The myosin heads, which have an ADP and phosphate connected, reach for the active site on actin, which creates a cross bridge. When the myosin head attaches to the active site, the phosphate is released. As the thin filaments pull toward the M line, the myosin head flexes. As the myosin head flexes, ADP is released and ATP attaches to the myosin head. Once this happens, the myosin head becomes energized at the expense of ATP, which is broken down into adenosine diphosphate, or ADP. This is at the expense of energy. Finally, the sarcoplasmic reticulum receives the calcium back. This calcium is attached to the calsequestrin, which allows tropomyosin to cover the active site again. This causes the muscle to relax after a hard day's work.